continuing with another great presentation by Dr. Maryia Trubitsyna. And before that, I would like to introduce her with a few words. Dr. Maryia Trubitsyna has a strong scientific background in molecular biology and DNA and RNA therapeutics development. She joined IQVIA in 2021 as a part of the strategy consulting EMEA group first. And currently, she's a consultant in genomic and precision medicines for Europe, the Middle East, and Africa at IQVIA. She's responsible for supporting development of the offers and capabilities of the genomics team, with focus on spotting and engaging with academic, private, and government opportunities in the region of Eastern Europe, Germany, and Italy. And today she will speak on the topic of making precision medicine a reality, innovations, trends, and challenges of the healthcare of today. So, Dr. Trubitsyna, without further ado, the floor is yours. Please share your presentation.
Thank you for the kind invitation and for inviting me to be here. It's a great event, and I'm sure we are all contributing to making precision medicine a reality by discussing it from different points of view. And today I'm going to talk a little bit about innovation trends, but also challenges that are stopping precision medicine to come to reality faster. And I didn't expect to have an introduction before my presentation, and I was going to introduce myself, as maybe I'm not that known as some of the speakers in this conference. My name is Maryia Trubitsyna, and I currently am in IQVIA Genomic and Precision Medicine team. My background is scientific development of gene therapy tools in the University of Edinburgh, where I was studying for several years novel enzymes to reduce the cost of RNA therapeutic synthesis, and it was before we all had mRNA injected in our arms, so it was a very innovative approach back then. And I also have some nice publications and author of 2 book chapters. My education undergraduate is in microbiology, and as mentioned at the beginning, I also joined IQVIA in a strategy consulting, so worked very closely with the small, large, and the top pharma companies, helping them develop the strategies for launch of the new therapeutics. And there is a small list of my hobbies in case you want to reach out as an icebreaker. So what I would like to discuss today and to present to you is the concept of precision medicine. What do we mention when we say novel therapies, and what challenges in the development and market access they are facing? I would like to talk a little bit about the whole genome sequencing for newborns, their opportunities, but also challenges and ethical questions. And then I would like to finish about the presentation about the concept of digital solidarity and datafication of our lives and growing up in the digital future. And my final slide will be about the readiness for digital health futures. So what is personalized diagnostics and personalized medicine? If we have a cohort of patients and the diagnosis is a particular type of cancer, and we give them a monotherapy, the same to the whole cohort, some subset of the patients will have a positive effect over time, some will have no effect at all, but small group of patients might develop the adverse event and sometimes even need to be hospitalized and admitted to the emergency settings. So personalized diagnostics is when we use additional tests to find the specific type or specific biomarker of the cancer type that they have. And we have personalized therapies. In this case, we are talking about cancer that will allow to get the beneficial effect in the whole cohort of patients. We already realize that one size does not fit all in the healthcare, and this approach is being applied in many countries nowadays. But what we can aim for is to be using personalized diagnostics to prevent disease, to enable early intervention and delay manifestation and progression of the disease for patients, but also for their family members who might be affected by the inheritable diseases. So precision medicine is not only about treatment, it also can be used to prevent, to assess the risk of the disease, as some diseases are monogenic, so one mutation is enough for disease to be progressing and to have manifestation in this patient, where many diseases have it will need several mutations to be accumulated, which increases the risk of development particular condition. So by having personalized tests and analyzing genetic information, we can assess the risk of developing particular disorder over time and estimate the probability of getting a disease. For diagnostics, precision diagnostics is used to end the diagnosis odyssey which many patients and families are undergoing specifically for rare and ultra-rare diseases. Because the clinical picture might not be so known for the diseases where there are a handful of patients in the country, and it takes around 80 years to diagnose a rare disease. Many families would rather have the answer straight away and spend these years in treating and easing the symptoms of the disease rather than finding the answers of what is going on. Genetic testing can allow to end the diagnostic odyssey and to have a clear answers and start the treatment as early as possible to prevent irreversible changes in the organism. And of course, precision medicine is used for treatment to identify the drugs, specific drugs, or even the dosage concentrations for different patients. And this is called pharmacogenomics, when the results of genetic tests are used by the clinicians to establish which dose of the drug will be the most useful for these particular patients. It's not only the genetic information which informs the precision medicine, it's also family history and also the lifestyle of the patients. And we already spoken a little bit about the innovative treatment, and what is included in this innovative treatment is cell, gene, and RNA therapies, which are called usually novel advanced therapies. And here I would like to mention a white paper that's been published in IQVIA Thought Leadership. So it's also, I printed maybe a little bit old-fashioned, but is nice sometimes to have a real paper as well. IQVIA Thought Leadership team prepared this white paper to discuss the promise of the novel therapies. They start from earlier on in 2010, when the first advanced therapies were approved, and then discussing what challenges the market access strategies brought to these therapies. In the 2020s, we're always going to be the era where the full commercial promise of cell, gene, and RNA therapies was to be realized. And also pandemic accelerated the development and uptake of mRNA vaccines, which are transformative for the whole field of advanced therapies. And to note here that before mRNA vaccines for the COVID pandemic, there were no approved therapies for mRNA. So it is a breakthrough for the whole field of RNA and DNA therapies. And here we are at the pivotal moment in the development of these therapies, where the next decade will bring enormous changes to usher cell, gene, and RNA therapies into pharmacotherapeutic mainstream. This decade, between 2010 and 2020, it was an era of steps forward as well as steps back in the progress to a viable commercial market for advanced therapies. Forward, because innovation was prolific and milestone approvals happened, but the early antisense RNA products were then later withdrawn, with the first cell therapies and genetically modified viruses, gene therapies, silencing RNA, and messaging RNA treatments. They brought breakthrough treatments for serious conditions later on. But some steps were back because commercialization of innovation was in some cases much less straightforward than anticipated and brought with it withdrawals and marketing discontinuation for some of them. And is a very good conclusion is the paper that the companies entering or already playing in the area, they need to be innovative not only in development of the therapeutics where they are, but also in their go-to-market models to realize the full success of the treatment. And the same as Sir Andrew Dillon mentioned, they have to remember that the novel nature of the therapeutics doesn't necessarily shield them from the competition of more conventional therapeutics where available. So up to date, there are 19 gene therapies approved, 17 RNA therapies, and 56 non-gene modified cell therapies. So if we are looking in the future, what is in the pipeline for development of the advanced therapies? There are around 3,600 therapies in advanced therapies in development. Around 2,000 of them are gene therapy, 800 cell therapies, and around 800 RNA therapies. The therapeutic area which are the top of the company's development is oncology and rare diseases. Mostly is this myeloma, lymphoma, leukemia, and ovarian cancer. In cell therapy is the same oncology and rare diseases, as well as COVID-19 complication. There is also development in arthritis and leukemia, myeloma, and ovarian cancer. In RNA therapeutics, preclinical development is dominated by RNA interference and messenger RNA and antisense therapeutic developments. For RNA therapeutics, the rare diseases and ultra rare are in the first place. When we are talking about RNA, DNA therapies, sometimes we operate the terms which are can be puzzling for general public, and in the next slide I compiled some facts which may be sometimes unknown and new to some people. We only know how DNA is organized, the double helix that is currently used everywhere in marketing, but we only found out how it looks like in 1952 by American biologist James Watson and English physicist Francis Crick. They came up with a double helix structure, and the DNA that encodes in our bodies, only 1% encodes for protein. So these are enzymes that make all other molecules in our bodies, and 25% of regulatory elements, and the rest so far is unknown. There are hypotheses of what this DNA can do. There are a lot of repetitive elements, transposons, and we are very similar to each other. So in the human genome, 99.99% is identical to each of us. And it's very common to say that because we are all built from the same 4 building blocks, A, T, C, and G, as all living organisms are, we are very similar even to our pets that sometimes we didn't think about. And there are 3 billion letters of code in the human body, where we're talking later about sequencing that generates very large files, and storage of information has to be that therefore specific, secure, and special tools needed for the sequencing files to be analyzed. And what is interesting that only in one cell of our body there are 3 meters of DNA. If we multiply by 15 trillion cells in a human body, all DNA together will be twice the diameter of the solar system. So going back to more serious novel therapies, I might be a little bit biased towards RNA therapies due to my scientific background, but also potentially to personalized search results in my browsing history, as everything now seems to be more personalized. Novel RNA therapies offer a wide range of solutions, and I mentioned some of these therapies already before: small interferon RNA and messenger RNA. If we start with CRISPR, in 2020, Emmanuel Charpentier and Jennifer Doudna received a Nobel Prize for a method for genome editing. So this system relies on a designed guide RNA and RNA-guided nuclease. And the first use of ex vivo CRISPR-based therapy to treat genetic disease, researchers treated individual in Germany in 2019 with beta thalassemia. CRISPR is now in clinical trials for blood disorders such as sickle cell disease. And if trial data continues to be as positive and it's been up to date, this treatment might be approved as soon as the next year. There are also more modern, more new development in direct DNA editing are base editors. If CRISPR makes a double-stranded break, base editors, they cut only one strand of DNA, which possibly have safer potential capabilities. The challenges of using CRISPR and base editors are off-target effects. So if the insertion is happening not in the place where we want to have, but to monitor this event, we have to do whole genome sequencing at different time points after the treatment. A little drawback of base editors is that currently there is only a possibility to change C to T or A to G. There is already first patient dosed with base editors for heterozygous familial hypercholesteremia in summer this year in New Zealand, and still 14 patients to be recruited. A small interfering RNA, there is also a Nobel Prize in 2006 to Andrew Fire and Craig Mella for discovery of RNA interference. And the RNA binds to the target mRNA and targets it for degradation. Thus, we can change the levels of the protein expression as well as increase or decrease them. And also, mRNA, as mentioned before, is being developed for personalized anti-cancer therapies, and the vaccines that already been a success in this and past year. So now I would like to discuss a little bit more in detail the challenges and the barriers that personalized medicine is facing. So on the left, I listed novel therapies: RNA, DNA, gene editing, direct gene editing, and cell therapies. The first issue is the specific tissue delivery. As these DNA and RNA therapies, they are water soluble, and our cell membrane are phospholipids, which makes it difficult for the cells, for these molecules, therapeutic molecules, to penetrate the cells. And there are many solutions that are in development. They are still in academic and R&D research, where exosomes are used. These are vesicles which are similar to lipid nanoparticles, but are naturally produced in our bodies from specific cell types. The spherical nucleic acid, DNA nanostructures, and very recently cell-penetrating peptide, the small peptides, they can guide the therapeutic molecules to enter the cells. As I mentioned also, for gene targeting, off-target effect for direct gene editing, it still remains the challenge, but hopefully base editors will provide higher safety levels for that. The production standards for cell therapy lead to the need for decentralized manufacturing, as personalized medicines need to be produced rapidly at very small doses because they are patient specific, and we do not need the large production lines or large fermenters to produce a large amount of this molecule, because it's only needed for a small subset of patients. The molecules that have been produced, if we're talking about RNA, DNA, they are quite stable. The cell-based therapy and the gene-modified cells are not that stable. If we take, for example, Strimvelis, this is a gene therapy for severe combined immunodeficiency, the therapy can be only administered in one clinic in Europe, in Italy, in Milan, and the shelf life of the therapy is 6 hours. So even not all tests and standards can be performed, and to find the bacteriological test of this therapy, you will only know the answer after the therapy have been administered, as it takes 24 hours to find out if there was any contamination. And of course, accessibility and cost. The cost of current CRISPR-based therapy is around 1 to 2 million US dollars, which can be performed in a very small number of medical facilities worldwide, putting it well out of reach for a vast majority of people in the world. In the next slide, I wanted to focus on the newest development of personalized cancer therapies, as the sequencing costs are going down, and we can now easily sequence the genomes of normal tissue and tumor tissue to obtain the information about new antigens that tumor cells can have. And then we can activate the person's T cells to recognize the tumor cells and to lyse the tumor cells. So far, there are only 3 approved personalized vaccines. The BCG live vaccine cannot be called that yet personalized, because it's a vaccine that's been used for over 60 years to prevent tuberculosis, and a couple of decades ago, it's been shown the positive effect on early stage bladder cancer. The exact mechanism is unknown, but possibly by infecting the cancer cells and increasing the immune response of the host. And there are also 2 vaccines for prostate cancer and melanoma that been recently came to the market. But also, interestingly, the exact mechanism of action is unknown, says the official page of one of these drugs. And as I mentioned here, the costs for the next generation whole genome sequencing is dramatically decreasing. And the advances in the technologies open up prospects to diagnose diseases and prevent onset of the diseases as a preventive personalized medicine by sequencing humans early on in their lives. So I wanted to present a concept, a dilemma of newborn whole genome sequencing, which is now discussed in the Genomics England. which diseases are going to be screened for, and who decides which diseases will be on the list. There is a set of principles that Genomics England, together with the steering committee, is discussing, but it's very good to see that the discussion is happening together with the involvement of the wider public, and anyone can comment or participate in the discussions, which diseases, how to choose the diseases, for which newborns to do the test, and what information to share with parents or with children at what age. As it's not that simple, that there are different diseases with the onset early on life, or it can the disease may not manifest until 30. So when this information should be shared and by whom? As some parents are not welcoming of knowing the disease if it cannot be curable, but maybe it's not curable nowadays and it will be in 5-10 years, and the child can be eligible for a clinical trial. We in the next year, Genomics England will be having the discussions with patient organizations, from affected families, and also together with the steering committee and the genomic clinicians to come up and verify the set of the diseases as well as the principles by which they are shortlisted. And generating large volumes of sequencing data creates next questions on how this data will be stored and where, how to ensure a lifetime safe environment, properly informed consent from parents and children, and of which age children can decide of how their genetic information is used. Access to such information has to be governed and protected at a national level. Talking broadly, data safety is not only relating to genomic data, as in the new world, a new term, datafication, is introduced, where our lives and activities in our lives is transformed into a digital form that can be collected, stored, and analyzed. Even before birth, very large volumes of information are collected about unborn child, started from ultrasound scans to parenting apps, online games, online watching history. And it might seem that the policies for we should aim that the policies and the governance for the data management develop at the same speed as the digitalization of the world around us. The Lancet and Financial Times Commission came up with the term of data solidarity, which is, along with equity, human rights, inclusion, and democracy, is one of foundational values that Commission believes should shape future governance in order to derive the greatest public value from digital transformation. Solidarity should be reflected in all dimensions of digital health, including collection, use, sharing of health data, and also data for health of the nation. Data solidarity approach safeguards individual human rights while simultaneously building up a culture of data justice and equity, and ensuring that value of data is harnessed for public good. And in my last slide, I want to mention that the Lancet and Times Commission came up with crystallized 10 key dimensions to measure the digital health future's readiness of the nation. There are 4 general principles that the human rights approach and the governance of health futures should be taken into account. The approach to all policies has to be people-centered, inclusive decision-making, not only for adults, but also taking into account views of youth and marginalized communities. Design that all people of all ages can access and understand the digital information and storage. That the benefit should be for all, that all households, health workers, facilities are connected, and also the digital health literacy in the general public to be increased. And the common health goals is doing no digital harm to the planet, and health and well-being should be a part of all digital policies. And that's the end of my talk, and there are a couple of minutes for questions. If you would like to discuss anything that I have mentioned today, please reach out to me, but taking into consideration the high error rate of spelling my name, take along my surname, there is iqvia.com contact, and please let know that this is considering genomic and precision medicine team. We will be more than happy to reply and discuss.
Thank you so much, Dr. Trubitsyna, for this great presentation. While the public leaves their questions in the chat box, I would like to ask a question myself. Because you mentioned a few times the access and the cost of these innovations in different countries across Europe and not only. So my question is, how can relevant stakeholders take steps towards a fair access and equitable access to these innovations in the following next years? Because we know this is a huge process that will take a long time. But in your opinion, what is the best approach and the next steps towards it?
I think the best approach is to be having conversations. So first start with a patient organization in the community to understand what are the needs and what is the most required and necessary for the patients to reach. And then it has to be an open dialogue between the specific community and the nation, country, and the pharmaceutical company. I believe that pharmaceutical company developing drugs for benefit of patients, not for their own benefit, and they are very keen and open to find new ways of how to find the solutions that are possible to get equal access in all the countries. And we see more and more nowadays with the different types of agreements between companies and government can be to solve exactly these questions, that solutions can be found by having a dialogue.
Perfect. Thank you so much. We don't have any other questions from the chat, but if any arise, I will send them to you via email later on the day. Thank you again for the presentation. It was a pleasure, and I hope to see you again in Biotech Atelier 2023.
Maybe in face-to-face event. Fingers crossed.
Hopefully. Yes,
Thank you so much. Have a wonderful day.